Yeah, so I'm going to talk about cell polarity and making the right number of polarity sites, and it's going to be biologically a story of two different fungi that made us think about what kind of models can we use if they appear to be doing things similarly but have different outcomes. Okay, so the motivating questions for me thinking about cell polarity are that, first off, question of how do you orient polarity in response to chemical cues? You can pick your favorite cells. This is just a small sampling, but a lot of cells will use chemical information from the environment to orient their activities. I'm going to talk about yeast. Cells are tiny, chemical gradients are often shallow and noisy, so actually this is not a trivial problem. How do you figure out which direction to go in? In many of these experimental systems, people have figured out what the chemical cues are and what the receptors are that are sensing those chemical cues. And once you've done that, you can do a very straightforward experiment, which is give the cells equal concentrations of a chemical cue coming from every direction. What do they do? And this varies a bit, but the general rule is that the cells polarize anyway, even when there's no directional information from the environment. And they look just as polarized as they would be if they had a clear direction from the chemical cue. So the chemical cue induces polarization. If there's a clear directionality to the chemical cue, it will polarize in the right direction. But if there's no directionality, it will polarize in some random direction and look just as polarized. So a second question is, how do they pick a random direction to orient all of their polarity molecules towards, and why is it only one direction when the information is coming from everywhere? Okay, so cell polarity is this kind of squishy concept, which means different things to different cell types. So in a migrating cell, there's a clear front and a back, whether it's a fibroblast or a neutrophil or whatever. But for example, a killer T cell has a front which is towards the cell it wants to kill because it's an infected cell. It's going to secrete all these killing molecules. It has to do it in the right direction. And epithelial cells have an epithelial and a basal side. Embryos, obviously, anterior and posterior. So in each of these cases, there's a different chemical cue that cells are interested in, and there's a completely different architecture that comes about as a result of polarizing.
You could have a neuron.
You could have a neuron too. Neurons are more complicated, so I just left them out. This is from a review that wasn't by me, but it was neurons do it too. So is there really any commonality here, since the cues are different and the outcomes are different? And there appears to be a commonality in the genetic circuitry that mediates this in most cases. And in the review, they highlight this protein called CDC42. There are relatives that have different names in other systems. It's a small GTPase prenylated at the plasma membrane. And what you would find in each of these cases is that whatever we would end up calling the front, that part of the plasma membrane is populated by the GTP-bound, the on-state version of CDC42 molecules. High concentration of CDC42 at the front, at the apical side, at the front of the crawling cell, at the contact site of the T cell. And the idea is that whatever the cues are that polarize you, they tell you where to put a high concentration of CDC42 in the plasma membrane, and then that instructs the cytoskeleton to do whatever is appropriate to that cell type's polarity. So the fact that there appears to be a conserved polarity circuit in between the cues and the outcome means that we can talk about polarity in general. That's something conserved. And then I'm going to skip over a lot of years of work in yeast to tell you what our conclusion was about what are the molecules. So we did genetics to find molecules, we did biochemistry to see what they do. And an entire field, but this particular model came out of my lab. And this is the critical set of molecules that we think are required for polarity to work and to be able to polarize spontaneously in a random direction. So what are they? The green one is an activator of CDC42. It's an enzyme that will load CDC42 with GTP, turning it into its active state. Once it is GTP bound, like all of the Rho family of proteins, it can be recognized by a set of proteins called effectors, one of which is called P21 activated kinase. Effectors are proteins that only recognize the GTP form, and then they do something when they bind to the GTP form. And then the third protein is a scaffold protein that binds 40 or 50 different proteins, among them these two. And what we think is critical is that it binds to both of these to the same molecule of BEM1. So we have a bunch of biochemical and genetic evidence for this, that forming a complex like this is critical for CDC42 to be able to polarize in response to cues or spontaneously. Okay, so I'm going to give you a cartoon. This is my biologist cartoon before we get to a mathematical representation. So CDC42 is a prenylated protein. I'm drawing it gray when it's off or GDP bound, blue when it's on or GTP bound. Because it's prenylated, it tends to diffuse on the plasma membrane. And this complex of 3 proteins I told you about is diffusing in the cytoplasm. Each of those proteins has a very weak membrane binding domain, but primarily they're cytoplasmic. There's another protein, the GDI, which is important, and I'm going to skip for simplicity. But what it does is binds the GDP form and plucks it on and off the membrane. So it's actually a mobility factor because it plucks one protein off of here, but it'll diffuse and plunk it on the membrane somewhere else. Okay. So with this set of proteins and the recognition that diffusion in the cytoplasm is much faster than it is at the membrane, which is actually quite extreme in yeast because the membrane diffusion is very slow.
I'm struck by the low value of diffusivity at the membrane.
Yeah.
It's almost like immobile.
Yeah.
Do you know the basis of that?
No, I don't. I'd say we don't. There are theories that it's due to the membrane composition of this particular membrane. I actually think of it as so in fungi, the plasma membrane is plastered against the cell wall, and there's multiple transmembrane proteins that are essentially immobile because of the cell wall. They're cross-linked into this one covalently linked molecule. So it's like a picket fence with a lot of immobile pickets, and I suspect that's contributing to this. But yeah, it's very slow.
Yeah, it's much lower than any transmembrane protein that we know of in mammalian cells, for instance, isn't it?
Yeah, and this is, if you look at the receptor, that's a 7 transmembrane receptor. We know the upper bound of the diffusion constant for that is 0.0005 micron squared per second. So it can get slower than this. Yeah, it's a very slow membrane.
And this is from single molecule tracking, or it's from?
This is mostly from FRAP, but there has been some single molecule tracking. Yeah, I mean, it's hard to do single molecule tracking over minutes, which is what you would need to measure this. Yeah. Okay. So, given this set of proteins, how would you go from unpolarized to a polarized cell? And how would you do it spontaneously so it could happen anywhere? And so here's our thinking. Suppose you're sitting in the middle of a cell looking at a patch of the membrane, not drawn to scale. You've got slowly diffusing CDC42s, and I didn't animate it, so sorry. But occasionally, stochastically, there will be a collision between the green guy and one of the grays, and it will turn blue. There are other proteins, gaps, which I didn't talk about, which turn the blues back into gray, and that's what most likely would happen to these blues. But one of them could get lucky and catch one of these complexes. Remember, the red guy binds to the blue. It's an effector. So this interaction is weak. It would, we think, last on the order of 1/10 of a second or a few tenths of a second. But that's long enough for that green guy to bump into a few of the neighbors and load them up with GTP. So then this complex falls off, but other complexes are likely to bind one of the blues or more of the blues and essentially amplify a zone of blue in a sea of gray. So that is the totally non-mathematical biological intuition we got from what we were doing. And a mathematician called Andrew Goryachev turned that into a set of PDEs. So these arrows here are all just mass action or the most straightforward linear biochemical reactions. And then you also have, of course, diffusion, which is slow for membrane-associated species, fast for cytoplasmic species. And here in the color bar, you have the concentration of the blue guys, this GTP-CDC42. And if I play that again, this starts in an artificial membrane with a single molecule or so in the middle. It quickly creates a zone of GTP-CDC42 and depletes components in the cytoplasm so that this stops the spread of the zone. So this, we think, is why a cell that's stimulated to polarize can take a starting noise and build itself a neighborhood of GTP-CDC42 around that random site. The problem with this is if you can start it with noise, why is it only one site? I mean, why don't you just start multiple sites? And of course, in the model, if you start it with 2 sites, you get 2 clusters. This is a no noise PDE model. Simulation is still going. No noise and symmetric, so that should not have happened. But of course, there's rounding error in the 8th decimal place. So eventually, this much less than biological noise is enough to create a tiny asymmetry between the peaks. And that deterministically leads to competition between the peaks, where one grows and the other one shrinks.
So is it is inherently bistable because of this BEM complex, which has some kind of nonlinear properties of recruiting more CDC42 and that.
Right. So if you imagine 2 clusters where one has a slightly higher concentration of GTP-CDC42, the stable cluster that you form is actually very dynamic in terms of the molecules. The molecules are coming in and out every 2 to 4 seconds. So if you have 2 clusters, anything that comes out is in a shared cytoplasm where a yeast cell is about 5 microns across. So with normal cytoplasmic diffusion, they can be caught by either cluster. And so once you have a slight asymmetry between the clusters, the one with more GTP-CDC42 is more likely to catch any complex that falls off of either of the clusters. Yeah.
These things, the 2 spots are like 2 peak arrays, is worse if they're actually very far from each other, because then there is both the poles can essentially become, let's say, can effectively become this. And the only way this process can, let's say this process of killing off one of the peak happens, it has to happen at the rate of through diffusion of particles from that peak to the other.
That's right.
And in a big cell, this can be a product, right?
Yes. So this in a big cell would be a different outcome, because this competition would be much, much slower if you have to travel very large distances they basically could be isolated from each other and maintained. That's true. But for normal size cells, so yes, if you're talking about an egg or something huge, but for normal size cells, diffusion in the cytoplasm is fast enough that this should work. Okay. So, yeah, the competitive nature comes from the probability that a cytoplasmic complex will be caught is higher if there's more local GTP-CDC42. And once you bind it, that dissociates, but if it dissociates in an environment where there's a high local concentration, it could rebind before falling off. So actually, the larger cluster is a bit better at recruiting. It's also a bit better at retaining complexes that might fall into the cytoplasm.
Kinetic resetting, when it unbinds, it will rebind with higher probability than.
That's right, depending on the local concentrations, right?
I think it has some similarities with what Alpha Yap had proposed for Rho, but with a different underlying kind of kinetic resetting that they proposed. Maybe we could discuss that.
Yeah,
it's
I'd
an
like
interesting paper with some theory.
Okay, yeah, I looked. Okay, so this is in the model. Is anything like this actually happening in the cell? So to do that, you have to get the movies working fast enough to catch these instances of competition, and we can do that. So one of those polarity proteins is in white. I don't know if you caught that, but there were transiently 3 that quickly shrank to 1. When it loops, follow this cell. There were a few transient complexes that shrank to 1, or take a look at this one. 2 complexes shrunk to 1. So this is actually not that hard to find once you can get the filming fast enough to see it. So the time scale is that the proteins are coming, if you do FRAP, the proteins are coming in and out in the 2 to 4 second time range. That's the half-life. Competition between patches is on the 1 to 2 minute time scale, which fits what, those dynamics fit what the model might predict. And then there are other cells where we don't see any competition. We just see one spot get brighter. Now, of course, we're missing anything that's below our detection threshold. So that we can't say anything about. Okay. So at least imaging is consistent with this idea that competition is happening due to basically a shared cytoplasmic pool being accessible to both spots. To ask if that's really what's happening in the cells, we did this experiment where we tried to change one of the parameters in the model. And the easiest one for us to change here that would make a difference is the off rate with which things fall off of that cluster, because the affinity of each of these components for the membrane is extremely low, and we know of other things that can make it higher, so we can attach a membrane philic piece to one of the cytoplasmic proteins. And now it will have a much longer half-life at the spot. And yeah.
Could you test on larger cells whether, there's a characteristic length scale, much larger than the normal wild-type length scale? So if it were larger, then you would see it.
Right. So we have, by making a mutant that cannot cytokinase, so it just gets bigger and bigger. Now, the shapes are rather weird, but those yeast will, after a couple of cell cycles, get big enough that now they can do 2 buds at the same time. So that is another test, yes.
So the length scale is like a few tens of microns, or?
I don't know that I can give you a length scale. It's a volume, I think, that we're... Right. Right. So once the cells get, let's say, 5 or 10 times bigger, volume-wise, than the starting cell, you can get 2 or 3 buds reliably.
Cytokinesis mutant would also have many more nuclei, right? So
it does also have many more nuclei, that's right.
So that your concentration...
The concentration is the same. We've measured that. But of course, the volume is much bigger. So yes, you have a lot more protein, which means since it's a cytoplasmic pool that you have to deplete, then yeah. Yeah.
So you showed multiple spots arising.
Mm-hmm.
If you just take a snapshot when there are multiple spots, is it always the case the biggest one wins?
In the strain that I showed you, the answer is yes, but that was a manipulated strain. and the reason, so in the wild type, the answer is no. I elided this, but in the wild type, there is also a negative feedback loop where this effector, the red guy in my cartoons, phosphorylates the green guy. Once you get a big spot, it phosphorylates the green guy and inhibits it. So actually, the dynamics in a wild type are rather more complicated. What I showed you was a movie from a mutant where I made a non-phosphorylatable version of the green guy so that we inactivated the negative feedback. So yes and no. Okay, so here what we did is just make the BEM1, the yellow component of that primer, more membrane philic. And the idea was, well, now the FRAP is slower. It's 20 to 30 seconds instead of 2 to 4 seconds. So it should take a lot longer for competition. And the cell cycle of the yeast is fast. So the cell cycle of the yeast polarizes and 10 minutes later starts budding. So they might start budding before the competition is done, and you might get more than one bud. And then, in fact, we can get that to happen. So in this mutant, it can make multiple buds at the same time. They're not the same size because they were in the process of competing with each other when they started budding. And this is, if you look at nuclei, this is a disaster for the yeast, because if you make more than one bud, now when you go through mitosis, you have 2 nuclei that you have to distribute 3 ways or 4 ways. And then this becomes problematic because as they fight for it, you become aneuploid. So these are very sick.
It's just a side question, but
yeah.
When you have, say, 3 different buds,
mm-hmm.
do you find that the total volume increase rate is the same as in whether with a single, just partition or smaller, or actually do you scale the
That's correct. What you just said is correct. So if a mother makes 2 buds, each bud is growing at half the rate than it would if it were making 1 bud. So the growth is limiting and it's split between the buds. Yeah. Okay. So in answer to that question I posed, what we came up with in studying Saccharomyces cerevisiae, I should have said this is Saccharomyces cerevisiae, the classical baker's yeast, is the reason that you can polarize spontaneously and have it be one direction is, number one, you have a positive feedback via this complex that amplifies starting noise to grow clusters of polarity molecules at random places on the surface. The clusters then compete for cytoplasmic complexes as well as 42, and a larger cluster will win in principle. Even with the negative feedback, it's always the case that the competition ends up with one winner. The dynamics are a bit more complicated. A larger cluster will win over a smaller cluster because it's better at recruiting factors and retaining them from the cytoplasm. Okay. So we felt pretty good about this. Yeah.
The simulation you showed, it was on a flat plane. Is there any?
Yeah, we had boundary conditions that connected the sides. So it was a torus in principle, but yes.
Is it anything that would be interesting for the curvature of the cell?
So Andrew Goryachev, this mathematician that I mentioned, has done that in a spherical cell or in an ovoid cell. It doesn't make a lot of difference to the outcome. Yeah. Okay. So a colleague of mine, and I should say the same circuit has been analyzed in fission yeast Schizosaccharomyces pombe by Sophie Martin's lab. And basically the exact same circuit appears to be at work there. And these are very distantly related yeasts. In animal cells, you have these GEFs called beta pics, where the GEF has attached to it essentially the yellow protein I had one, so that it will bind to pack molecules. So the same complexes are present in mammalian cells. And we thought, okay, this is universally how it should work. And then this colleague, Amy Gladfelter, who was sampling marine fungi off the coast of Cape Cod and putting them on the microscope, sends me a movie and says, you have to work on this. This fungus doesn't care what you think. And, and it's disobeying everything you said. And so this is a fungus that makes multiple buds at the same time routinely, and it makes decisions that we don't understand about how many buds to make. It doesn't die. Unlike cerevisiae, it has multinucleate cells to begin with. So when you make multiple buds, you can still give everybody a nucleus. But it does seem to care that it doesn't make buds for which it would not have enough nuclei. So if you look at, these are now all Arabicidium pullulans cells, but the ones with only one nucleus look just like Saccharomyces cerevisiae. They only ever make 1 bud. Now, if you have 2 nuclei, generally you make 2 buds. You can also make just 1 bud, and you can make 3. In all those cases, you still have enough nuclei. If you have 3 nuclei, you tend to make 3 buds. 5 nuclei, it's not a 1-to-1 relation, but the modal category of how many buds you make is the number of nuclei you have to begin with. Yeah.
The nuclear position itself biases the components that need it.
Yeah, so how does a mother know how many buds to make? We actually started with 3 theories, one of which is yours. So the hypotheses we had were, okay, one, if you have more nuclei, you're bigger. And with Turing systems, there's a characteristic wavelength. And so maybe the number of wavelengths you could fit into the cell would somehow scale. So size seemed like the most obvious way to do this. Alternatively, like you said, it could be the number of nuclei, like each nucleus has some kind of connection with the cortex. And so you're counting number of connections, and that tells you how many buds to make. Less obviously, it could be just the total amount of DNA in the cell that somehow affects transcription of something, whatever. So those were starting hypotheses. Of course, all of these things are very highly correlated with each other. The DNA is correlated with the number of nuclei 1-to-1. The size correlation is not perfect. This is if you plot the number of nuclei and what size you get, there's quite a range, meaning that for any given size, you can have quite a few different numbers of nuclei. And particularly down when you get here, 1 and 2 nuclei, there's a lot of overlap. So if you can find very easily about half the cells overlap. So you can find in the overlapping zone what are the rules. And when you have 2 nuclei, you tend to make 2 buds. And when you have 1 nucleus, you tend to make 1 bud. In fact, you always make 1 bud when you have 1 nucleus. We have seen one instance in about 3,000 cells where it made 2 buds. But you basically make 1 bud if you have 1 nucleus. And if you have 2 nuclei, half the time you make 1 bud, half the time you make 2 buds. And there's no size difference between the ones that make 2 buds and the ones that make 1 bud, statistically speaking.
Maybe the spread in the, for a given volume, you said you could have different number of nuclei, but because this is a growth process, if you plot time on top of it, what I mean is that for a given amount of time that you spend since you've had 2 nuclei, the volume is increased an amount. So the spread of values is not some kind of random noise, but rather reflecting growth, isn't it?
Yeah, that's a good point. We have tried to correlate timing as well as size and still find no correlation. So there's a correlation there because bigger cells tend to be born with 2 nuclei instead of 1 sometimes. So there is a correlation, but time also doesn't explain what's going on here. So this suggests at least if size could be a factor, but it's not a determinative factor. So we're saying no to the size. So then how do we distinguish number of nuclei from amount of DNA? Obviously they're linked. Yeah.
But I guess you're saying cell size is not a parameter that is sufficient to account for.
Correct.
But it is nonetheless possibly a necessary.
A contributor, yes. Yes. How do you separate these? Well, in principle, you could separate them if you made different ploidy nuclei, right? So a diploid nucleus would have the same DNA as 2 haploid nuclei. What would its behavior be like? So Avril, my student, and this is the person who's been working on this fungus, she used the genetic trick borrowed from Saccharomyces to double the ploidy. And you can, by flow cytometry, show that you've doubled the ploidy. And now you can take a look at the cells. And this is a haploid and a diploid. And the haploid, of course, makes 1 bud. And the diploid, in general, makes 2. And one of them gets left without a nucleus. So the diploid population ends up throwing out massive amounts of dead cells that have no nucleus because they can't go anywhere. Okay, so we don't think it's the number of nuclei. We think it's the amount of DNA, which is the hardest and least attractive of the 3 hypotheses that we started with. Okay, so how does the amount of DNA affect the number of buds? This is our theory. The amount of DNA somehow gets translated into maybe a different level of some transcript. And that transcript maybe is a regulator that changes something about the polarity circuit so that you get a different number of buds. This is total hand waving, of course. Yeah.
But if you had a critical molecule whose amount in the cell is set by the amount of DNA. What I have in mind is what Jan Hartman proposed for cell cycle regulation.
Yep.
Could remember the name of the genes, but
We5. Yep.
Exactly. It's as a constant amount when the cell divides, and I think it's scaled by the amount of DNA. Couldn't that set a rule?
Right. So that's essentially what we're thinking about. Is there something that it's not actually okay? We've done proteomics on haploids and diploids at this point, and the vast majority of proteins are present at the same concentration. But there are a set of proteins that are at different concentrations, and almost all of in fact, all of the statistically significant ones are higher in the diploid than in the haploid. So There are proteins that do differ. We don't yet know which of them is important, if any, but those are our candidate lists, right? So, yeah.
Just doubling, you make two birds, and you, as you pointed out, that there are a lot of them end up without a nuclei.
Right.
Then is it fair to say that maybe just that double the amount of DNA being split into two nuclei prevents or reduces the chances of there being a bud which doesn't have any DNA? Not contributing to the number of buds, but it is kind of contributes to making sure not one of them is dummy.
Yeah, no, I think in terms of what Thomas was saying, in terms of the why of it, yeah, that the cell does not want to make cells that don't have nuclei. They're a dead end. They're not helping the population. They're not Darwinian fitness. So yeah, the whole point of counting DNA in what is normally a haploid, unless you've tricked it, is to not make cells that don't have nuclei. Yeah.
Yeah, has anyone in the lab done like kind of a modeling forward approach where you take the yeast model where you can find one polarity site and just from a concentration dependent way, can you create multiple stable sites? Because I guess you described how just like decreasing the diffusability of the, and just decreasing competition allows for multiple polarity sites. But if you just increase the concentration of any components, does that?
Yes. Okay. So that's where I'm going. But I'm not going to do it. So Goryachev's original model, I think, had 8 PDEs. Our model with the full circuit has at least 10, and it's a massive simplification of what's probably really going on. And I don't know of any way to sweep parameter space for something with that many parameters to figure things out. So we tried going minimalist, going to simpler models first. And this is what the family of mass conserved activated substrate model. So if this is kind of the model, the mechanistic model that I was telling you about with real proteins, many people have tried to simplify this to a minimalist model that has the key ingredients. And the key ingredients are an activator that's got positive feedback to make more activator, and a substrate that can diffuse much faster than the activator because it's in the cytoplasm. So you can think of this as the Rho GDP or the CDC42 GDP form and GTP form. It's not really that because each of these species has to account for behaviors of multiple proteins. But it gives you a much simpler model. And they're called mass-conserved activator substrate models because there's nothing in here about synthesis and degradation. And the reason for that is, we've done experiments, for example, where you can shut off synthesis of any of the polarity proteins. And the cells are actually fine for a few generations until they dilute out what they started with. So polarity can happen without synthesis. And they're very stable proteins. They get diluted when cells proliferate, but they don't get degraded at an appreciable level. So, and the time scale of polarization being, a couple of minutes is really fast relative to the yeast cell cycle, which is 90 to 120 minutes. So for that reason, yeah. Okay, so for that reason, we assume that there's no synthesis and degradation, that there's a conserved amount of this U plus V entity. And that gives you an easy to handle set of models where U locally will change according to diffusion and some biochemistry that we have to talk about in a minute. And the GDP form will change by diffusion and a biochemistry that's linked by mass conservation.
This is presumably where you will have this substrate depletion.
That's correct.
That is critical for the whole thing to work.
That's right. That's right. So the U will get depleted. The V will get depleted, sorry. And then, of course, diffusion in the cytoplasm much faster than the memory. Okay. So here are the two most popular ones, but by now there have been on the order of a dozen different versions of F. So this one is what Goryachev came to as a simplification of his original model, and it's been called the Turing style model. This has, so what you can see is a positive feedback that's nonlinear in the U squared, and a V there because you can only make more U by converting V to U. And then the reverse U falling off the membrane to make V is generally modeled as linear. Okay, and then for this one, you have a saturable positive feedback. And then this gives what's being called the wave pinning model. But regardless of which of the dozen or so models people have come up with, all of the biologically realistic ones, and by that I mean ones that don't allow the concentrations of U of V to go negative, for example. So all of the biologically realistic ones have certain features. One is that as you add more and more protein into the cell, you reach saturation of the peak of U, because the valley of V basically saturates at some very low level. So at some point, your peaks start getting broader rather than taller in terms of concentration of U. And if you look at what happens to the different models when you start out with two unequal clusters, there's, in theory, three possible outcomes, right? If the bigger one does better than the smaller one, you will end up with one, and they will compete, and this is the Saccharomyces cerevisiae model. If it doesn't matter, if they're basically all fine with each other and they're sharing, and then whatever you start with can be what you end with, and there's just drift. Or alternatively, you can imagine a situation where the bigger one loses faster than the smaller one, and then the smaller one would win until it becomes a bigger one, and so you would get equalization. So just theoretically, there are three classes of outcome. And what can be shown for all of the minimalistic models, no matter what the FUV is that we've analyzed, is that they always yield competition. Now, initially, I was kind of shocked by this because the papers, papers coming out of a mathematician who proposed wave pinning did not show competition in simulations. And the ones coming out of the Turing model did show competition in simulations. And I'm telling my mathematical collaborator, but this doesn't fit. And we argued for a while until I realized there was a language barrier between the biologist and the mathematician. Mathematician is, it will compete on the time scale of the age of the universe. I don't care what you think. And the biologist is like, no, but I mean biological time scale. Oh, well, that's you. So when the peaks get saturated, they complete slowly. In 1D, they compete on the time scale of the age of the universe. In 2D, they actually still compete on reasonable time scales. But biologically, this would be like the cells that I made with slow competition, where you'd make different bugs. Okay. But the always competition doesn't seem to fit with what we're seeing in this organism. I didn't tell you, but the black here is a fluorescence on a polarity marker. And it looks like they equalize. And if we film them making the polarity sites, they can start out quite different, but they'll end up the same before they start budding. So it looks like this guy is doing equalization, even though every model we analyzed was doing competition. So, yeah.
I'm struck by the fact that at least in the towards the left,
Mm-hmm.
the buds grow at the region of highest curvature. Is this something that you systematically analyze? What I have in mind is like K on K off, we depend on available membranes to volume
Right.
surface ratio, and that does it tune the kinetics?
So, okay, I didn't say this, but there are membrane-bound upstream regulators that can enhance the probability of polarizing there. And so places where you have budded before are particularly favorable for budding. I don't think it's curvature, but I don't know if that also affects it. Yeah.
So two different diffusion rates kind of already makes the membrane bit of a sink because things don't get out of that place as much as they come in. So is in the species that where there's more buds being formed, the migration form, is the membrane diffusion rate in the membrane even slower, or is it this comparable?
I think it's comparable. We haven't systematically done FRAP on cells of different size, but my impression is it's comparable. Okay, so I think this organism is doing equalization of different numbers of sites, and yet none of our models do that. So these models are too simple. We have to go to something a little more complicated than a two-component system. It turns out that you can get there with just one more component. So this is what we call an indirect substrate model. It is exactly the same as the UV in that model, but there is a third species, which is something that comes off of U and can be converted to V, but only with some delay. An indirect substrate. This was inspired by what some of the actual proteins do that we look at in the much more complicated model. And the equations you can see here. So we're starting, let's say, with a Turing version where you have positive feedback that's nonlinear, linear off. You turn that into this simply by having another linear off that gives you the indirect substrate. For V, you have the same as before, plus another linear term coming from the indirect substrate. And the indirect substrate basically gets made by coming off of U, consumed by turning into V, and also diffuses at some rate. And if you do that, this three-part model can give you equalization. And the way it does that is, so if you start out with two peaks of different height, and now these are just different, slightly different parameter spaces in terms of the total amount of U plus V plus indirect substrate. So going from less to more. You have less, then as in the two-component model, what happens is this. A larger peak is better at depleting locally the cytoplasmic concentration. And so you generate a gradient in the cytoplasm, small but present, from the neighborhood of the small peak towards the large peak, which is why competition gets driven, right? Molecules fall off here, they diffuse net this direction to be caught by the large peak. That's how competition works. Once you have an indirect substrate and the peaks are large enough and they're beginning to get saturated here, now what happens is you've released a species that isn't immediately sucked back up by this peak. It goes a little bit further out. So instead of being down here, it actually makes the neighborhood around that dip higher. And that turns the net cytoplasmic gradient in the other direction, which gives you equalization. Okay, so with this model now, you can do, it's simple enough that you can map out some parameter spaces, and there's parameter spaces that will give you competition, equalization. Coexistence is where competition happens, but it's slow. Okay, so yeah.
From the point of view of the data, how would you tell if there's like equalization or just they both reach independently a concentration? If there's a maximum concentration that they can reach.
Right. So that, so that's a great question. On occasion, you can actually see a situation where the smaller peak and the larger peak gets smaller as the smaller peak gets bigger. That just tells you can transiently populate a larger peak than what you end up with. But I don't know that answers your question really. It's also.
If you see the larger peak shrink, then that would say that it's equalizing.
Okay, then it's equalizing. The reason I'm not sure is if you instead of tweaking the mass conserved activated substrate model, if you go to a not mass conserved model, then yes, you can get independent peaks whose synthesis and degradation locally will sustain themselves, right? So I'm reluctant to say we've shown which of those is true. Okay, so with these simple models, what we can say is that in a minimalist model, there's always competition for a single winner. In a more complex, similar type model, you can get different outcomes depending on parameters. And so now the job is to go back to this guy and figure out what it's doing with its polarity circuit and what the parameters are that we need to look at. I will also say, if you relax mass conservation, even a small amount, Erwin Frey has shown that also can lead to equalization. So there's other ways to do it than by having another component. Okay, so what does the polarity circuit look like in this fungus, this Arabicidium pullulans? It looks like it has two parallel circuits. So CDC42 and RAC are different names given to basically very similar proteins. So it has two of them. In fact, most cells, most animal cells, most fungal cells have two, at least, of these family members, CDC42 and RAC. And what we've shown is that the same activator, GAF, activates both of them, and the same GAP inactivates both of them. And oddly enough, if we delete one or the other of these circuits, you can still bud, but the number of buds changes. If you delete this circuit, you get more buds for the same number of nuclei. And if you delete this circuit, you get fewer buds for the same number of nuclei. I can't tell you what's going on, but this is the current state of the art on where we are. It has two polarity circuits. They seem to have intrinsically different numbers of buds that they prefer to make. And when you put them together, the cell counts appropriately for the number of nuclei that it has. Okay, so that's it. What I told you about in terms of the experimental stuff with Arabicidium is all this wonderful MIT grad student, Avril Wang. And the theory was done by Jiangang Chu, who now has his own lab in Taipei. And this is my lab at MIT. Thank you for your attention. Yeah.
So when you told us about the equalization circuit, I thought you were going to tell us that maybe it would also oscillate sometimes, right? And I was wondering, there must be like an over dampedness to that. And I was wondering if you could get rid of, in order to test some of these hypotheses, if there's any way that you could, I can't tell off the top of my head, slow down or speed up something in order to get oscillations. But that might lead to oscillations if you didn't over dampen.
Right. So in Saccharomyces, what happens is you get competition and there's a single winner, and that happens before that one starts oscillating, which happens afterwards. In pullulans, the initial polarity circuits start oscillating before they equalize. So I don't know what to make of that yet. But one of we think that one of the key oscillatory factors is this protein PAC, and we can delete that. And now what we get is uneven polarity sites that make uneven buds and don't oscillate. So I don't know. This is where Thomas was saying if we'd gone in for the genetics and characterizing phenotypes, we would not understand what we were looking at. So we don't understand what we're looking at, but that's a phenotype.
And so you don't know how to engineer it so that it oscillates, or do you?
Sorry.
Do you understand how you would have to tweak the parameters for it to oscillate? Do you predict an experiment?
We can add a negative feedback where the local concentration of this and this factor in so that this inhibits this, which is what happens in Saccharomyces. And if we do that, the circuit will oscillate. We don't know what's going on in this new fungus. Yeah.
Just curious, so when those proteins form clusters, so does that lead to any sort of significant change in the local membrane curvature that in turn might encourage, clustering, have some sort of positive feedback?
Yeah, well, yes. There's a 10-minute delay between when you cluster. So the proteins cluster, then they organize actin cables. The cables bring in vesicles. When you fuse enough vesicles, you get a change. Because this is all under turgor pressure where the membrane is flattened up against a cell wall, and you need to actually remodel the cell wall before you'll get any curvature change. So there's a significant delay between the dynamics I'm describing and when you actually get a curvature change to make the bud. But yeah.
Presumably, if you increase the diffusivity of CDC42G-TP by some experimental means, you could see regimes of traveling waves of excitability. Do you ever see that in simulations, in like an experiment to test it? I mean, so long as you don't have any depletion of the substrate that would prevent that.
Right.
Obviously.
So I don't know how to make something diffuse faster. I mean, we've tried raising temperature, but that what we've done that sometimes gives you this kind of behavior is if we get rid of actin, and then cells can't bud, they just get big. And if we do it at a stage of, we arrest the cells at a stage of the cell cycle where they're not terribly keen on budding, but they would be okay with mating, which is another case where they have to polarize, then you can get patches that travel around the cortex. In fact, we think that's important for cells to find each other when they're searching for mates. Okay, here. Oops. So these are two cells that would like to mate, but the polarity sites have formed in the wrong place. So they're pointing in the wrong direction in order to mate. And then they start moving them along the cortex. So they meet and then they stop moving.
That's what happens when they mate.
That's right.
Related to this, do you know then if the fusion rates are really the main difference between the system here in the yeast and in other systems where the waves have been shown? Like there's this interesting work on the sister egg. In sister egg, we have all of these patterns of weird waves and stuff that form.
Yeah, I mean, the circuitry is very similar, right? It's the same kind of model. So yes, I think that and also, in terms of the instabilities you'd see if you had two clusters, it's not just this competition I've been talking about, but if the fusion was faster, you would see merging, right? That's the other form we don't see that in yeast. In fact, even if the clusters are right next to each other, they'll compete, but they won't merge. And I think that's because of this incredibly slow diffusion. Yeah.
Have you done any experiments where you replace the Orobasidium genes with yeast or with the Saccharomyces genes, and does that really change?
We have tried a couple, but it turns out that because of codon usage and other things, we need to be cleverer about how we do that than we have been. Doesn't like Saccharomyces genes. Yes.
Just a comment, which is that because it's a random connection, but could potentially be useful, which is that this patterning is actually also exactly what you see in shear banding in amorphous solids. So, and there's a lot of literature on that. And the way to see it is if you take a chunk of material and you do this to it, like there's fluctuations. And once you form a shear band, there's a process by which when you dissipate work, you generate more plasticity and local, anyway, you get a shear. And it's exactly the same in the sense that once you saturate, there's a global pull, which is the total strain in the whole system. So just to say that in case it's useful, because there's people who have thought about that process a lot in the context of that problem, and I think it's nearly identical mathematically. So just in case, because if you start to form two shear bands, if the thing is small enough and the global strain is accommodated quickly enough, you only have one. But there's a time scale where if you shear it too fast, then you actually form two, and then two can saturate. So I'm just saying, like, in case it's of use, like this is sort of
I would love to.
There's a competition between shear band and these systems. It is very similar mathematically.
Cool. All right.
I mean, I feel a bit embarrassed that I don't have a good understanding of why equalization emerges from the existence of this B1 into
the indirect.
Yeah.
Substrate?
Could you explain to us not just the mathematics, really kind of the intuition behind it, why it works?
Okay, so
I can explain it to someone else.
I don't know if this will work, but I'll try. Okay, so imagine you have a small cluster and a large cluster. The large cluster is really good at catching the substrate, but it can't do anything about the indirect substrate because that's not yet in the form that's catchable. Okay, so it's releasing, if you just have the two-component model, the larger cluster is releasing more substrate and catching more substrate. With the indirect substrate, it's releasing a total of indirect plus direct substrate that's higher than what it can catch because it takes time for the indirect substrate to convert to direct substrate. And then if that indirect substrate can move away a little bit in that time before it converts to substrate. Now what you've done is created the reverse gradient. You've got a substrate that is now higher in the outskirts of the big peak than on the outskirts of the small peak. So you have a net cytoplasmic.
So in parameter space, what is critical is the value of the off rate to V1 with respect to what?
Well, so the parameter space that I plotted is actually just the diffusion constant of the indirect substrate. So this is the total amount of U plus V plus VI on the y-axis, and the diffusion constant of VI. So if it's too slowly, it converts back to substrate right under the first peak, and you get competition. And if it diffuses fast enough, the other way to think about it, if it diffused infinitely fast, what this would be is like you degrade some of the U from the peak, and you synthesize V everywhere, which is also a way to make them independent and reach the same. Yeah. Okay. Thank you.